Okay, so this is chapter 3 in your book. We're skipping chapter 2. So chapter 2 should be a review. It's basic chemistry and like macromolecules, that kind of thing. So we're going to chapter 3. So you are going to learn a lot about gram staining. So the person that developed gram staining is Hans Christian Joachim Gram. Okay, so they keep calling it Gram. Okay, so he was a physician. He worked at a morgue, and he worked with this person, Friedlander. He was trying to figure out what actually caused pneumonia. So Gram was coming up with ways to stain and actually visualize bacteria under the microscope. So he's trying to figure out how can we see these things? So one thing that he noticed is with one of the methods he developed, the bacteria didn't stain the same way. So some were pink and some were purple. So basically some retained the dye, hung on to it, and some didn't. So that actually revealed that there were two different kinds of bacteria, which we call gram positive. So lots of times we just say G positive or gram negative. So you will do this in lab and repeatedly over and over again. Okay, so first thing we're going to talk about prokaryote versus eukaryote, which was the last thing I talked about in the last set of lecture notes. Okay, so prokaryote, remember, these have no membrane-bound organelles. Right, so that means no nucleus, no Golgi complex, like all that's time. Think about all that stuff you spent all that time learning about in biology. Prokaryotes don't have them. Okay, so the two examples of prokaryotes are bacteria and archaea. Okay, eukaryotes, okay, they have membrane-bound organelles, so they have a nucleus. I am going to ask you this, so make sure you have this straight in your head. Okay, so eukaryotes are fungi, animals, plants, algae, so protists, so basically everything else. Okay, now we actually, in treating disease, we can exploit similarities and differences between the different cell types. So we actually, some antibiotics, okay, so these are effective against bacteria. Okay, they are effective against prokaryotes. So what we do is the antibiotics can affect the prokaryotes. So they're working on basically the differences between the pro and the eu. So they affect the prokaryotes without having a lot of side effects in us. Okay, so you should definitely know the basic difference between the prokaryotes and the eukaryotes. So it's all about membrane-bound organelles. Okay, now we're going to go over all the prokaryotic cell structures. Okay, so let's just start with the surface layers. Okay, so I have a picture here. It's easier to kind of do it with the picture. Okay, so let's start with the cytoplasmic membrane, and then the cell wall and a capsule. Okay, so the cytoplasmic membrane is basically the same as lots of times we call it the plasma membrane or the cell membrane. Okay, so this is the phospholipid bilayer that goes around the cell and basically regulates what comes in and out of the cell. So controls cell transport. Okay, now I'm writing this down and there's more of it. So this is kind of like the introduction. So, so this is an introduction. So I'm not going to write on here anymore. We'll do it on the next slides. Okay, so here's the stuff on the outside, right? And this is the dividing layer. And then we have cytoplasm and then nucleoid, which we're going to talk about in a minute. Okay, so if you look at this picture, this is a very typical bacterium. Okay, so here is this brown, that's the cytoplasmic membrane. Okay, and then bacteria have a cell wall most of the time. Okay, so there's the cell wall, and the cell wall is going to be rigid. Okay, now some bacteria have an outer layer, which is called a capsule, which we'll talk about. Okay, they have flagella, not all of them, but some of them have flagella to move. They have these cytoplasmic extensions called, an individual one is called a pilus. Okay, and we will talk about those and what they do. Okay, cytoplasm is the material in between all of the stuff in the cell. Bacteria generally have one circular chromosome, and it's in a region that's called the nucleoid. Okay, so now this is an electron micrograph. So this is really fine detail, and you can kind of see some of this stuff. So you can see the cytoplasm, you can see the cell wall, the nucleoid region. Okay, but now we're going to go over all those parts. Okay, so the cytoplasmic membrane, it defines the boundary of the cell, and it's really going to regulate, and I don't have enough room down here, regulates what enters and exits the cell. So cell transport. Okay, so same idea as in general biology, right? So it's a phospholipid bilayer. So that means there's two layers. One has a or both layers look the same, but they're kind of mirrored. So there's a hydrophilic head and hydrophobic tail. The hydrophobic tails want to be together, right? And not in the water. So basically it kind of looks like this. So this is the hydrophobic part, this is hydrophobic, and then hydrophilic is out here. Okay, it moves. Okay, so it's fluid. It's not stiff and rigid, it's fluid. Okay, now we call it mosaic because there are proteins integrated on the surface and in through the plasma membrane. Okay, so all of these things that are different colors, those are pretty much proteins. Okay, so some of them are going to be sites for things to bind. Some are channels, which we're calling gates. Some might be receptors, some are enzymes, right? So there's all of these different things in the plasma membrane of the cells, and all those proteins have a function. Okay, now, bacteria and archaea have the same general structure. Okay, so they have, their phospholipids are different. The archaea, the tails aren't fatty acids, so they're hooked up differently to the glycerol, so they're not fatty acids. I'm not going to ask you to draw this or anything, but they are different. Okay, now, efflux pumps, which in biology, we never call them efflux pumps, but efflux is kind of like exiting. Okay, so these are transport proteins that are going to move waste or toxic products out of the cells. Okay, so some bacteria have efflux pumps that get rid of the antibiotics that somebody's taking. So they pump it out and then they might be able to survive. Okay, so this is maybe a form of antibiotic resistance, or why one antibiotic will work against one bacterium and not another one. Okay, so these efflux pumps, so basically it's cell transport. Okay, now active transport, I don't know that I need to go over this again. Active transport, remember, it requires energy. So energy, we say lots of times it's ATP, right? And active transport, we're going to move against the concentration gradient. So we're moving from low to high. So normally with diffusion, right, that is, we're going from high to low. So that's kind of like the laws of physics. Things move from a higher concentration to a lower concentration. So they just, that's the nature of like chaos. Okay, so they move from a place where they're more organized to a place where they're less organized. Okay, now active transport means we're taking it in the opposite direction. Okay, so we're going to go from low to high. So sometimes we're using ATP, but other times we're using the power of a whole bunch of protons. So it's almost like going over a waterfall. The water's going to carry it through. So this picture shows facilitated diffusion, where things are just going to go through a channel, but they're still going high to low. Okay, then we have active transport, where we're going low to high. Okay, then active transport using ATP. Okay, so the ATP down here is driving this pump. Okay, so it's making it happen. And then we have group translocation. So group translocation, it's going to alter the transporter, chemically alters the substance as it's transported across the membrane. Okay, so these are just different ways that things are moved across the membrane. We will not spend much time after this on this. Okay, so group translocation is common in bacteria. Okay, so we take it across and then we alter it, we change it. So as the compound is moving through, this protein that's in here is going to change it somehow. So one way that we change it is phosphorylation. So with phosphorylation, what we're doing is we're adding a phosphate. So bacteria, when they're bringing a glucose molecule into the cell, as it comes through, it's changed a little bit. And what they're doing is the bacteria are adding a phosphate to it. So it's going to make that easier for the bacteria to actually use the glucose with the phosphate once it's in the cell. We will talk about this more as time goes on, but there are certain proteins in bacteria that actually get excreted out of the cell, and we call them exoenzymes. So exo, exit, so they're external to the cell. So if this is the cell can make these enzymes. Okay, and they actually get transported out of the cell, and they actually function and work out of the cell. Okay, so they're in here, they get excreted, and they work outside of the cell. So it's interesting because sometimes bacteria break down molecules out of the cell, and then the molecules get broken down into these smaller components that can then be absorbed into the cell. I always think about this like a mama bird. So like if you think about how the mom feeds the baby bird, so the mom eats the food, digests it, regurgitates it into smaller pieces, and then those smaller pieces can get absorbed by the baby birds. The baby birds eat the small ones. So we will talk about specific exoenzymes as time goes on, okay? But just be familiar with the general concept. Okay, now this is where it gets interesting. So we're going to talk about cell wall. Okay, so the cell wall is kind of rigid, okay? And what it's going to do is it prevents the cell from breaking, from bursting or lysing. Okay, so sort of rigid. It doesn't have anything to do with cell transport. Okay, so it's going to keep the water from running into the cell or water from leaving the cell. So just it's like this outer wall. Okay, now, two main types of bacteria. So we're going to have gram-positive and gram-negative. Okay, so what makes gram-positive and gram-negative different is the amount of this chemical, which is called peptidoglycan. Okay, so peptidoglycan is drawn down here. Okay, so peptidoglycan is these two glycan chains that we put together. Okay, so they're sugars, they're chains of sugars basically. So these purple and blue things. So these chains of sugar. So they N-acetylmuramic acid and N-acetylglucosamine. Okay, so chains of those. So we have those in rows like this. Okay, and then those are hooked together. And what they're going to be hooked together with is actually peptides. Okay, so they're kind of hooked together like this. Okay, so if you look at this, so here's the glycan chains, so the sugars. So think of this as a sugar. Okay, and then in here, these are amino acids. Okay, so those are the building blocks of peptides, right? So protein, think protein. Oops, I spelled it wrong, peptide. Sorry, I could erase, maybe. Where's my eraser? Uh-oh. Sorry, here it is. Okay, so let's fix that. Okay, so peptides in here. All right, so it's kind of hard maybe to get, but if you look at the word, okay, so peptido, so that's the protein part, and then glycan, that's the sugar part. Okay, so it's these sugars kind of in parallel lines. Okay, and then those parallel lines are going to be connected together with proteins. So I read an example one time where they talked about drift fence. Okay, so you know the orange fence that like either farmers put up or they put up around construction sites. And the orange drift fence kind of looks like this, right? So the peptidoglycan is kind of like if you had rows, which I'm not going to be able to draw this, but rows of drift fence, like all lined up parallel, and then those are connected. So kind of like this. So this would be if you were looking down from the top. Okay, now gram-positive cell walls have a thick peptidoglycan layer. Okay, so here's the plasma membrane or the cytoplasmic membrane, right? And then out here, they have a very thick layer of those proteins and sugars. So they have a thick peptidoglycan layer. Okay, they have this thing called periplasm, which is the stuff in between, kind of like cytoplasm. And then they also have these things called teichoic acids. And teichoic acids are a place where things can hook onto it. So like here is a cross section down here. So here's the inside of the cell, plasma membrane. Here's the periplasm, which is kind of space. It's got some stuff in it. And then here's the cell wall. Okay, now gram-negative cell wall looks different. Okay, so the gram-negative cell wall has, I'm going to start on the inside. Okay, so here's the plasma membrane. Okay, here is the peptidoglycan. Here's periplasm again. Okay, gram-negative have another layer of phospholipid bilayer. Okay, so they have another phospholipid bilayer. So they have another membrane. So another lipid bilayer. Okay, so then on the outside of those, they have this stuff that's going to be really important, and it's called lipoplasmin. lipopolysaccharide. Okay, so lipopolysaccharide we're going to talk about in a minute. Okay, so the difference between this, right? So thin peptidoglycan, and then another plasma membrane and lipopolysaccharides. Okay, now lipopolysaccharides are also called LPS, and they're also called endotoxins. Okay, so endotoxin. So now, gram-negative bacteria, if they are broken apart when they die, okay, they release this endotoxin, or LPS. They release it, okay, and it goes into the bloodstream. And what happens is that this LPA can trigger a very extreme immune response, and it's called endotoxic shock, or would be kind of like septic shock if someone is septic. Okay, so what this LPS does when it gets released into the bloodstream is it causes the blood vessels to dilate. Okay, so that means they're going to get big, right? Blood pressure is going to drop, and somebody's going to go into circulatory shock. So they're not going to have enough blood circulating in their body. It's going to pool. So that can happen when gram-negative bacteria are killed really fast. Okay, so and it's this endotoxin. And the endotoxin is the same as the lipopolysaccharide. That's what it is. It's just another name. Okay, now I don't know if the gram-negative cell wall, that outer membrane part, makes them less susceptible to certain antibiotics. Okay, so some molecules can cross by holes, but they don't necessarily get killed. So different antibiotics work against gram-positive than gram-negative sometimes. Okay, now there are antibacterial substances, so basically antibiotics in a way, that actually target that peptidoglycan layer. So the classic example is penicillin. So penicillin interferes with the formation of peptidoglycan. Okay, so it stops them from being linked together. So that's going to kill like gram-positive. So they're usually more effective against gram-positive than gram-negative. They've modified some penicillins, and that makes them broad spectrum, so they'll work against both. But the original penicillin is more effective against gram-positive and gram-negative because of the thicker peptidoglycan layer. Okay, now another thing that's an antibacterial substance that targets peptidoglycan is lysozyme. So you might have heard of lysozyme. So basically, it's also called lysing enzyme. So you find it in tears, saliva. Okay, so basically what it does is it breaks down peptidoglycan. So it's one way that our tears protect our eyes from bacteria. Okay, so it's basically considered part of our immune system. So part of our innate immune system. All right, now some bacteria don't have a cell wall. So you should know this as an example. So mycoplasma are a type of bacteria, so they're totally different. And they don't have a cell wall. So penicillin isn't going to work against it. Mycoplasma actually causes walking pneumonia, which we'll talk about when we get to lung diseases, but they don't have a cell wall. Their membrane, their plasma membrane has sterols in it, so it makes it tougher. So they don't need a cell wall. They grow really, really slow though. But Mycoplasma is a type of bacteria that doesn't have a cell wall. So you can't gram stain them. Okay, now the archaea, they have different cell walls. They have they don't have peptidoglycan. They have a thing they call it pseudo-peptidoglycan. We're not really worried about this, so you don't have to memorize that. Just know they're different. Okay, now on the outside of bacteria, so outside the cell wall, okay, some bacteria have a layer outside of that, and it can be called a capsule or a slime layer. Okay, so usually they are sugars and they're kind of sticky. Okay, but some of them are proteins. Okay, so they say a gel-like layer outside the cell wall, and it helps protect the bacteria that's inside there, and it also makes them sticky so they can attach to something. So like if it gets in your nose, it could get in your respiratory tract and stick because of this capsule on there. Okay, so capsules, you can usually see they're very distinct. Kind of like in a negative slide, you can see the capsule, but then some of them have a more diffuse, irregular kind of layer around them, and they call those the slime layer. Okay, now biofilms are layers of bacteria that have capsules or slime layers, and they just kind of stick together. So biofilms are just layers of bacteria. So one good example of a biofilm is like when you wake up in the morning and your teeth feel kind of filmy, that's bacteria and it's a biofilm. So they're kind of all stuck together. You could also see it sometimes like the dog water bowl or the cat water bowl. If it doesn't get washed all the time, right, you just keep putting water in it, it'll develop like a slime layer in there. And that's actually bacteria that are just kind of forming and they're sticking to each other. So biofilms are really important because the bacteria communicate differently if they're in the biofilm, and it can be harder to kill them if they're all packed together. So biofilms are interesting, and they'll keep coming up as the semester continues. Okay, now flagella. Flagella, you guys, I think know. So flagella are kind of like a string-like structure. They're used for motility, right? So that's how the cells are going to move. Some of them are important in disease. So like H. pylori, which causes stomach ulcers. Okay, so it's important for the movement of that organism. There are words to describe. So there's vocabulary to describe the arrangement of flagella on bacteria. So peritrichus, so trichus is hair, trich is hair. So peritrichus, it has the hairs all around them, the flagella all around them. Polar would mean they're at one end. Atrichus, none, there aren't any. And then lobopho means they have them on both sides, kind of like ponytails or pigtails, I guess. All right.